Welcome back. We're going to keep going on membrane lipids just for a little bit here. And I want to give you an idea of how we know some of the things that we know about cells and cell behavior and things. So that's what we're going to do here. We'll look to see how people know that there are all these different types of phospholipid and how we can analyze different types of phospholipid distribution in cells. So let's back up a little bit. Previously, we talked about a technique called centrifugation, and we talked about differential centrifugation and things like that, where you can fractionate cells. So when you do that, you can actually isolate the membrane part of cells. So you can pellet cells at a certain speed, or you can lyse cells, and you can lyse them with certain detergents to just isolate the membrane components. And we can also then think about how we might do that to isolate just the mitochondria, or just the lysosomes, and then isolate the membranes from those structures and start to look at the different types of phospholipids and the relative proportions that we see in those structures. And that's going to give us a little bit of a clue about the structure of the membrane and therefore what functions might be happening on that membrane. So before we look at just a technique very quickly, let's just kind of review. Here's our phosphoglyceride, here's our phospholipid, here are two hydrophobic tails, there's our glycerol in pink that we're familiar with, and then here I'm just showing you the different head groups for three different types of phosphoglyceride: phosphatidylethanolamine, if we put this ethanolamine here, choline, so we make phosphatidylcholine, and then serine. So you can see that these groups are all charged. They'd be charged at certain pHs, obviously, and not charged at other pHs. And so these groups have a certain charge, a certain polarity that we could use that as a chemical property to separate those different lipids. So let's imagine we've just taken some cells growing in a dish, and we've got those cells into a tube, and we've broken the cells open, and we want to isolate just the plasma membranes. So we do our kind of differential centrifugation thing, so we isolate just the membranes. Then we can use a technique called thin layer chromatography. So we can isolate the lipids from the membranes that we purify from the cells, and then we can separate them, and we can study the different groups of lipids using a technique called thin layer chromatography. And this involves this thin layer plate. So this is a thin plate often made of ceramic and coated with a material, and then we can use solvents such as acetone and chloroform. So if you've done a general biology 1 lab about photosynthesis, you might have done an experiment where you separate pigments using this kind of thin layer chromatography, except you use a piece of paper and then you have a solvent of acetone and chloroform, and you separate the different pigments based on the polarity. The same thing is going on here. So we're separating the lipids based on the relative polarity. So what we do is we take our plate and we make a concentrated spot of the isolated lipids at the bottom of the plate. And then we put the plate into this little bowl of solvent, and the solvent is just below the origin spot of lipids. And so as the organic solvent moves up the plate, the way that this type of plate works is that the phosphatidylserine is going to be retarded by the organic solvent, and the phosphatidylethanolamine and the cholesterol will be rapidly solubilized in the solvent and moved down the plate. And so we can separate out the phosphatidylcholine, phosphatidylserine, the choline, the ethanolamine, and the cholesterol based on their relative polarities. And this is all to do with how the polar head groups in each of these phospholipids and the single hydroxyl and cholesterol interact with the silicic acid in the plate. And so cholesterol, if you look at cholesterol, it's only got one hydroxyl. It's the only polar functional group, and so it's really not polar at all, and that separates all the way up here. And if you looked at the relative polarities and calculated the relative polarities of these phospholipids, then we would see that phosphatidylethanolamine less polar than phosphatidylcholine, more and less polar than phosphatidylserine. So we get this separation. You can then scrape these individual fractions from the plate and analyze them to look at chain length in the fatty acids and or carbon number in the fatty acids, that kind of thing. So that's it. Just a real quick taste of how we can study membrane phospholipids to get an idea of the diversity of lipids that we find in the membrane. I'll be back soon.